Lipids are liquid as well as solid fats, and people kind of assume that lipids are non-polar because they're not soluble in water, but lipids can actually be polar or non-polar. So the non-polar lipids, this includes triglycerides and something called sphingolipids, whereas the polar lipids are phospholipids. These are found in cell membranes, and these are called glycerolipids. And in terms of the fat we digest, where it comes from, well, it can either be endogenous, which is bile, or exogenous, which is dietary fat. And first of all, looking at dietary fat, there's three forms of dietary fat. There's triglycerides, which make up 90% of the dietary fat, and this is glycerol with 3 fatty acids joined by ester bonds. Phospholipids, which are glycerolipids, which are the polar ones, because they have glycerol with 2 fatty acids and a polar head group. And finally, cholesterol and lipovitamins, which makes up 5% of the dietary fat. And the cholesterol is either esterified or unesterified. Looking at the endogenous fat, which is the bile, what's the fat in the bile? Well, it's phospholipids, which is phosphatidylcholine. It's unesterified cholesterol, membrane lipids from desquamated cells, and lipids from dead bacteria. And I can also mention here the enzymes required to break down these three things, the triglycerides, phospholipids, and cholesterol. So lipase enzyme is to break down the triglycerides. Phospholipase A2, PLA2, that enzyme deals with phospholipids. And carboxyl ester hydrolase deals with the cholesterol. As a really sort of overview of the fat digestion, essentially this is what happens. You've got the lipids in the gut lumen. They're digested by lipases to break down the triglycerides, and the products of lipolysis, which is the breakdown of the lipids, diffuse through and enter the enterocyte. Fats are first mechanically disrupted, so physical breakdown, and this happens in several different steps. Food prep, chewing, and gastric churning allows mixing with digestive enzymes from the stomach. Squirting into the duodenum using the pyloric sphincter of the stomach. Intestinal peristalsis mixes the luminal contents with pancreatic and biliary secretions. And this is all mechanical disruption, so food prep, gastric churning, squirting into the duodenum, and intestinal peristalsis. Mechanical disruption, what's the purpose? Well, it's to increase the surface area to volume ratio to make it easier to digest and absorb the fats. Fats are also stabilized as an emulsion, because as we know they're not soluble in, well, the majority of them are not soluble in water, so they need to be stabilized as an emulsion to be absorbed. So you've got the small lipid droplets as a result of the mechanical disruption, and they're coated with bile salts from the liver, which includes cholesterol, lipids, and proteins. And essentially these are polar molecules. So what they do is that the polar head groups of the bile salts, they stick out to mix with the water, and the non-polar tails stick in, and the inside contains what I'm going to talk about is triglycerides and cholesterol esters. And this prevents coalescence, which means the lipid droplets sticking together, prevents that from happening. This forms an emulsion, and the core contains triglycerides and cholesterol esters. Fats are first digested by lipases from the salivary lipases and gastric lipases. So let's have a look at that. Well, lingual lipase digests the fats, then gastric lipase from the stomach digests the fats. And what happens? Well, the triglycerides, the glycerol with 3 fatty acid chains, it's digested into diglycerides. So the 3 fatty acid chains, one of them comes off, so it forms a diglyceride and a single fatty acid. And then what happens is that these lipases, they're inactivated by pancreatic proteases. So proteases, they digest proteins. So the protein enzymes from the pancreas, they inactivate the gastric and lingual lipases. And then what happens is, so these fatty acids, they can either be long chain or short chain. Now the long chain fatty acids, they're insoluble at a low pH, and therefore they remain in the core of this lipid droplet with bile salts on the outside, with the polar head groups out and the non-polar tails in, and the long chain fatty acids remain in the core of this. However, you also have short chain fatty acids, and what happens is that the low pH due to the gastric acid, they ionize the short chain fatty acids, and therefore they become soluble because now they're polar, I guess. So they're soluble, and they're absorbed across the mucosa and enter into the portal blood, which goes to the liver, as we know. And as I said, the lipases are inactivated by pancreatic proteases, and I've written here cystic fibrosis with an arrow and a cross. So cystic fibrosis somehow prevents the inactivation of lipases. So let's see how that happens. Well, if you recall back to the lecture on the pancreas, you'll remember that cystic fibrosis is a gene, it's a mutation in the CFTR channel, which prevents, and because of this mutation, chloride ions cannot leave the ductal cells, and therefore they can't enter back into the ductal cells through the chloride bicarbonate exchanger, and therefore bicarbonate and water are not secreted from the ductal cells. And if chloride and bicarbonate are not secreted from the ductal cells, then the pancreas produces thick protein secretions. And these thick protein secretions block the ducts in the pancreas, leading to pancreas destruction. So cystic fibrosis leads to pancreatic destruction. And if you destroy the pancreas, then you lose the proteases that the pancreas makes from the acinar cells. So you'll have less lingual and gastric lipase inactivation, because, as I mentioned a second ago, it's the pancreatic proteases that inactivate the lingual and gastric lipases. So therefore, if you have cystic fibrosis and you lose these proteases, you will have extended fat malabsorption slightly, but not overall. Well, why not overall? Because if you destroy the pancreas, then you're also losing the pancreatic lipases. So overall, you're not going to get increase in fat absorption, but cystic fibrosis will alleviate this effect slightly. And we can see that here. So cystic fibrosis extends gastric acid activity, alleviating fat malabsorption slightly. Now, the emulsification that I mentioned by the bile salts from the liver, that happens in the duodenum because, so in the duodenum, fat is emulsified by bile and digested by lipases from acinar cells of the pancreas, so pancreatic lipases, right? So let's see how that happens. Well, fat reaches the duodenum and it stimulates the duodenal eye cells. Remember, eye cells are associated with fat. And eye cells of the duodenum, they produce something called cholecystokinin. And if you think about it, if to absorb the fats, you need to create the emulsion. So cholecystokinin somehow is going to cause the secretion of bile. And how does it do this? Well, it causes the gallbladder to contract and it causes the sphincter of Oddi to relax. So the gallbladder contracts and the sphincter of Oddi relaxes. It allows the secretion of bile through the common bile duct into the major papilla of the duodenum, and that bile forms the micelles for emulsification, right? And if you have fat in the duodenum, you also want to digest the fat. So cholecystokinin, it stimulates pancreatic acinar cells to secrete pancreatic lipases and esterases. And what the pancreatic lipases do is they digest all of the remaining dietary triglycerides. Now, why have I said remaining? Well, if you remember back to the stomach and the lingual lipases, the gastric and lingual lipases digested 15%, approximately 15% of the triglycerides to make diglycerides and fatty acids. So the pancreatic lipases and esterases, they digest all the remaining dietary triglycerides.